Okay, our next video clip here, we're going to review some key features of proteins and nucleic acids, those last two types of organic macromolecules. All right, so this is a diagram from your textbook, which is trying to give you a reminder or a feel for the different levels of structure of proteins. So you want to read and review that information in your textbook if you haven't done so already. So proteins can have 4 levels of structure. They all have 3, but some have 4. So the first level of structure is called the primary structure. And the primary structure, so what are the building blocks of proteins? We've already said those are amino acids. So your primary structure is the amino acid sequence. So most living things use 20 different types of amino acids to build their proteins. So there are 20 of these smaller amino acid molecules available in cells that can be linked together in chains to build different types of proteins. There are some exceptions in the microbial world where some use 1 or 2 more. So you put these together in a sequence, and you can imagine there's a lot of variation. You've got 20 to choose from, and you can put those together in different lengths, different orders, and that's going to allow you to create many different kinds of proteins with their own special properties. And then once you have your chain of amino acids, and we get to chapter 8 later in the semester, we'll learn more about how all of that works. Then you get to what's called the secondary structure. And so these amino acids, what they do, some of them repel each other, some of them are attracted to each other, some of them love water, some of them hate water. And because of that, that kind of dictates a 3-dimensional structure that's beginning to take place within that protein. And so these little chains of amino acids, they form little areas of what we call local structure nearby other amino acids within this protein that's being built. And those secondary types of structures have names like beta pleated sheet and alpha helix and random coil. So if you hear those terms being used in a discussion about proteins, that's what's being referred to. So like a beta pleated sheet, it's like a flat surface, but it's pleated like a sheet. It kind of zigzags like that. An alpha helix, a helix is a spiral type shape. So those structures form locally. But then really even more importantly for a particular protein is what we call its tertiary structure, that 3rd level. And that's where it truly folds up into its 3D shape. I'm sorry, it looks like I'm writing like a kindergartner on the screen. They have not found a stylus that can write on the iPad that I'm using to annotate these slides in a legible way. So the tertiary level, that's the 3-dimensional structure of a protein. And it's that 3D structure of a protein that almost always dictates what its function will be. Proteins can have lots and lots of different types of functions, and we'll talk about many different types of proteins as we move through the semester. So some proteins exist in cell membranes and help move things across membranes. Some proteins are chemical signals that are used by living things. Many proteins are enzymes. So enzymes are little protein machines that carry out chemical reactions. Toward the end of the semester, we'll talk about antibodies. Those are protein weapons that your immune system makes to help you fight off infections. And the list goes on and on. Proteins a lot of times are referred to as the workhorses of cells. And then finally, that 4th level of structure is called quaternary. Come on, stylus. Quaternary structure. And so some proteins like hemoglobin, for example, if you've taken biology 202, you have multiple proteins that have folded up into their tertiary 3D structures, and they get together with each other and make a more complex type of protein. Not all proteins do that, but the ones that do are said to have quaternary structure. So another term to be familiar with too, all right, so you're thinking about chains of amino acids. What are some other terms to be familiar with? Because we'll see some of these used. Peptide, polypeptide. Those terms also refer to chains of amino acids. A peptide is usually a short chain of amino acids, 6, 8, 10, 12, 14, and so forth and so on. So you'll see that term used. And then polypeptide is a longer chain of amino acids. And sometimes polypeptide is used interchangeably with protein. Some people will refer to a polypeptide as just that chain of amino acids. And then once it folds up into its 3D structure and actually has a function, then it becomes a protein. But just get used to all of this terminology: peptide, polypeptide, and protein. All of those things are referring to chains of amino acids. All right, how about nucleic acids? So nucleic acids, that's our fourth group of organic macromolecule. And some examples of nucleic acids are DNA and RNA. And over here, this structure that you see over here is kind of a generic diagram of a DNA molecule. This little tiny structure you see there is supposed to be a generic nucleotide. So DNA and RNA are built from nucleotides. Those are the building blocks of these types of molecules. And if you look carefully at that figure, a nucleotide has three major components. It has a phosphate, it has a sugar that's called a pentose sugar, and a pentose sugar is a sugar that has 5 carbons in it. And then finally there's a nitrogenous or nitrogen containing base. And so what are our nitrogen containing bases? Those are adenine, guanine, cytosine, thymine, and uracil. All right, abbreviated A, G, C, T, and U. Which ones do you find in DNA? These 4 you find in DNA, A, G, C, and T. In RNA, you have U's in place of T. In RNA, you'll have uracil in place of T. So these nucleotides that have A's, G's, C, T's, or U's in them are linked together in chains to build DNA or RNA molecules. And DNA molecules are typically double-stranded, so you have 2 chains of A's and G's and C's and T's that are paired with each other. So we just said a nucleotide over here has a phosphate in it, a pentose sugar, and then one of those nitrogen-containing bases. So to link those together in chains, you have something over here that's called the sugar phosphate backbone, right there and right there. What that means is the sugar of one nucleotide is bonded to the phosphate on the next, and that creates the actual backbone of our DNA or RNA chain. So that's going to be true for both DNA and RNA. Then you've got the A, G, C, or T if you're DNA. If you're RNA, you're not going to have a T, you're going to have U's instead. And if you notice in DNA, it's a double-stranded molecule. And the A's on one strand are paired to T's on the next. If you got a C over here, it's paired to a G over here. A G over here is paired to a C over here on that side. And the reason those are attracted to each other is A's can share hydrogens with T's. G's have hydrogens in them that can be shared with C's. That means those hydrogens kind of float back and forth between those nitrogen-containing bases. And that's a type of chemical bonding that's not as strong as covalent bonding, but it's strong enough to help hold these DNA chains or strands together. And then finally, those DNA strands, so you've got this pairing that takes place, and then the molecule becomes twisted, and that's why we sometimes refer to DNA as a double helix, because you've got 2 strands of DNA, 2 DNA chains, and then that structure is twisted into a shape that's called a helix, and because of that we refer to it as a double helix. RNA over here is usually single-stranded. So again, you've got these nucleotides, A, G, C, and U, because you don't have T's, and they're all bonded. The phosphate on one is bonded to the sugar of the next, and that helps hold them all together. All right, what are the key functions for nucleotides? These are your information molecules. And that's kind of the key thing to think about with your nucleic acids. DNA, that's where your genes are located. Your genes are blueprints. They're genetic blueprints, chemical blueprints that generally tell your cells how to build particular proteins. And we're going to cover that when we get into Chapter 8 later in the semester. RNA molecules play different roles in transmitting that type of genetic information. And again, we're going to see that when we get into Chapter 8. But again, these nucleic acid molecules are the information molecules, absolutely critical for living things to survive. All right, there is a very special type of nucleotide called ATP. You hear about this in any biology class. ATP stands for adenosine triphosphate. And here is a bigger diagram of what ATP looks like. So we just said nucleotides are the building blocks of DNA and RNA, and all nucleotides have a phosphate, a sugar that's a pentose type of sugar, so it has 5 carbons in it, and then there's the nitrogen containing base. So ATP actually has 3 phosphates in it. There's the 3 phosphates. That's why it's called triphosphate. A phosphate is just a phosphorus atom surrounded by oxygen atoms, so bonded to oxygen atoms surrounding it. Those phosphates are attached to the sugar right there. It's a sugar that's called ribose. And ribose is a type of pentose sugar. It has 5 carbons in its backbone. And then that sugar is attached to our nitrogen-containing base. Okay, and in this case it's adenine. In ATP it's adenine. So that diagram there is showing you what an adenine actually looks like in a little bit more detail. And remember, there are some carbons in there as well. It's an organic chemistry type of diagram, and they've left the carbons out for simplicity's sake. All right, so where does this adenosine name come from? Adenosine is a term that refers to adenine bonded to the sugar, the ribose sugar in this case. You put those two together and you have adenosine, and then you add the phosphates to the adenosine, and now you have adenosine triphosphate. ATP, this is the fuel, I can't write today, fuel or energy molecule for cells, their preferred fuel source. You talk about that in any biology class. There's energy stored. If you look here on the, let me clear all this. If you look here on the diagram right there and right there, they've got a little squiggly line that indicates that those are bonds that are broken by cells, and that gives off energy. I know it's kind of weird to think about, but energy is stored inside the chemical bonds of molecules. You break those bonds and that energy gets released, and then the cell can harness that energy and use it to power things that the cell needs to accomplish, like moving things across the cell membrane or building things. Anything that requires building is going to use up a lot of energy to make that happen. All right, and as we'll see as we move farther and farther into the semester, so we just talked about how like carbohydrates are the preferred energy sources for cells. Lipids can also be used as energy sources, and so can proteins. As you guys know, you can get energy from proteins as well. What cells do is they break those larger molecules down, they harness some of the energy, and then they store that energy by creating these ATP molecules. It sounds like kind of a complex process, but these ATP molecules are molecules that cells can immediately or instantly use as energy sources. So again, sounds like kind of a convoluted process to take these other molecules, break them down, harness energy, and then store that energy in these ATP fuel molecules, but that's the way it works with living things, because these ATP molecules are energy molecules that cells can use instantly or immediately. And again, we're going to talk a lot more about that when we get into Chapter 7 in Unit 2. For now, just think of ATP molecules as the gasoline of cells. All right, the last lecture for Chapter 1 before we move on to Chapter 2, we're going to talk a little bit about naming and classifying and identifying microorganisms, and so that'll be the subject of the last video lecture for Chapter 1.